Hi again, everyone. Welcome to Chapter 13, where we're going to talk about sexual life cycles and in particular a mechanism known as meiosis, which is one of the key mechanisms for the major function of sexual reproduction, which is the establishment of genetic diversity. That's a theme that we're going to touch on throughout this lecture. One of the major characteristics of a living being is the ability or the potential ability to reproduce. Whether it's a direct copy, what we would call asexual reproduction, or involving more than one parent and sexual reproduction. The ability of the DNA in our cells is part of that mechanism of reproduction. It allows traits from ourselves and our progenitors to be passed on. And the genetics is the study of those mechanisms, how we inherit characteristics and how those characteristics might change based on circumstance or over time. The idea of heredity is that most of the traits, most of the functions, the vast majority of traits and functions are going to be handed down to the next generation. On the other hand, there are mechanisms of variation, whether they are random mechanisms of variation, or in the case of sexual reproduction, there are non-random mechanisms of variation. And so when children are produced from a mother and father, we can often see in society how the traits of both the mother and the father can be seen in the children. That is to say, they don't inherit the physical traits. What they're inheriting is the genetic material that gives rise to the traits. And so we have not only the preservation of characteristics, but also the intermixing and the increase in variability. When it comes to genetics and genes, we have terms that are very, very old, and we have terms that are newer. The concept of the gene as a unit of heredity actually predates our understanding of DNA as the heritable material. We knew that traits were passed on from parents to offspring, but we didn't really have an understanding of the mechanism. And so we have some of these older terms mixed in with newer ones. And so genes can be the units of heredity. That is to say, these are the mechanisms by which traits are inherited. So these genes are passed on across generations through the reproductive cells, which we often call gametes. That is to say, sperm and egg are the two gamete cells. Genes do have specific addresses on chromosomes. There are examples of disease, and if you look in the case study that we studied recently, we show you the Philadelphia chromosome, where genetic material from one chromosome has been exchanged with another. And we have actually changed the location, which is where this word comes from. Locus means location or address. We change the genetic material on those chromosomes, and that leads to a change in genetic expression. But under normal circumstances, genes have very, very specific localities on a given chromosome. Chromosomes are made up of DNA and protein. And so when we look at a chromosome, and these are mother and father chromosomes, these are what we call homologous chromosomes. Let's say this is chromosome 21 from the mother and chromosome 21 from the father. We would find the genes that control certain functions and certain traits to be in the same location on either chromosome. So what's happened is that over time we've gotten very, very, very good at mapping out where genes are. And so these numbers on the left are like a zip code. They say, okay, XP 1122, P meaning this is the P arm and this is the Q arm. This is the centromere of the chromosome right here. And so these give a specific address. And now we're starting to match up certain types of genes and or the variations of the genes that lead to certain types of disorders and diseases and so forth. So let's talk about the differences between sexual and asexual reproduction. In asexual reproduction, what generally happens is you make a copy of the genes from the parent to create an offspring, which is what we call a clone. That is to say, it's a different individual, but it has genetically identical material. So a clone is an individual with a genetically identical genome. For instance, in the War of the Clones from Star Wars, there was a single bounty hunter who donated his genetic material, and then they made clones of that person to make an army. But we can see this in everyday life. When we have identical twins, they are genetic clones. In sexual reproduction, the idea of cloning is relatively rare. It does happen from time to time, but what we have going on in sexual reproduction is that we have unique combinations of the genes. You're getting only a subset of the genes from each parent. You're not getting all the genes. In asexual reproduction, you're getting all of the genes from one parent. In sexual reproduction, you're getting an asymmetric set of genes from both parents. Now, human sexual life cycle is relatively straightforward. You are created from a fertilized egg. It's called a zygote that develops into a fetus and then eventually gives birth to a baby and a child. Then you reach sexual maturity and so forth. Other organisms undergo a more complicated form of sexual reproduction. That is to say, they may have a sexual cycle and they may also have an asexual cycle, that they adopt the characteristics of both asexual and sexual reproduction. Remember that the fundamental goal of sexual reproduction is to introduce genetic variability. A little secret about organisms that undergo asexual reproduction. There are some that, there are some complex organisms that will do it. But most of the organisms that undergo asexual reproduction are things like bacteria and so forth. And what's interesting is that they sort of introduce variability when they are copying their DNA. The rate of mutation in an organism that undergoes predominantly asexual reproduction is higher than the mutation rate in an organism which is a sexually reproducer. So they introduce variability in a different direction. In human beings, since we are sexual reproducing organisms, during human development, during the development of the human body, we actually set aside a special population of cells that are devoted solely to creating our reproductive cells. The other set, the one that make all of our tissues and bones and muscles and so forth, are known as somatic cells. These cells do not give rise to reproductive cells. They only give rise to other somatic cells. So in a strange little way, human somatic cells reproduce asexually. They have 23 pairs of chromosomes, 22 homologous chromosomes, and then a pair of sex chromosomes, either XY or XX under normal situations. So a typical animal cell, a somatic cell, will have those 46 chromosomes. We can look at those chromosomes, especially when they are pre-mitotic. Right before when they're pre-mitotic, we can actually analyze them visually, and we call that a karyotype. And in normal cells, we would see a pair of each of the 22 autosomes, homologous chromosomes, and a pair of sex chromosomes. So a homologous pair of chromosomes have the same length and same shape and generally have the same banding. We'll look at some pictures of chromosomes here in a little bit, and you can see the banding, and usually they have similar banding patterns. But again, sex chromosomes are considered, we call them sort of homologous, and they are directly homologous in a female. that's XX, but our non-homologous pair in the XY. So those are called the sex chromosomes, and generally they are determined by either the XX or XY. And it would be an interesting discussion to talk about the fact that we really do have non-binary situations. We are certainly binary in the majority, but there are people that are X, XY, and other combinations of sex chromosomes. So females in human beings are XX, males are X and Y. And again, there are some variances, and that can lead to some very, very interesting and powerful discussions about the binary role of our sex chromosomes and sexuality in human beings. Those other 22, those non-sex chromosomes, we call autosomes. You might hear out in the world or down the line, you might hear about an autosomal dominant disease or an autosomal recessive. What they mean is that the genes involved in that disorder are located on the autosomes and not on the sex chromosomes. An example of a sex-linked disorder, Fragile X is an example, or another sex-linked one is a sex-linked color blindness, for usually red-green. So in humans, we have a total of 46 chromosomes. We inherit 23 from the mother and 23 from the father, and our cells are what we call diploid. That is to say, the N here stands for the number of different chromosomes that we inherit from each parent. So when you inherit 23 from the mother, 23 from the father, we have 2 times 23, 46. Other animals have a different N number. For instance, in a horse, it's actually 32. They have 64 chromosomes. In the case of a dog, it's more like 40 or 58 chromosomes. They have 39 pairs. Okay. Frog, it's a little bit less than that. They have 11 pairs, so 22 chromosomes. So 2N for the frog would be 2 times 11. 2N for the dog would be 2 times 39 in the horse and so forth. And so we're going to be using representations like this, and you should get familiar with looking at these representations. For instance, these chromosomes here have been replicated. Chromosomes do not normally look like this. We have homologous pair here, but again, this cell is about to undergo some form of division, whether it's mitosis in a somatic cell or meiosis in what we call a germ cell. A somatic cell and a germ cell. We can't tell from this picture what's going on yet, but in any case, these chromosomes are getting ready to undergo a split of some kind. It just depends on what kind of cell we're talking about. So in the human life cycle, before fertilization, you have 2 cells that are haploid. The sperm cell that has 23 chromosomes and an egg cell which has 23 chromosomes. They unite and fuse, sharing the genetic material to create a diploid zygote. So a zygote is a term for a fertilized egg. The zygote will eventually begin dividing, and dividing very, very quickly by mitosis. So once we have had that fertilization, the cells are either going to undergo mitosis, a form of, shall we say, asexual reproduction to create the multicellular organism, or we'll have meiosis to create new reproductive cells. So that cell will be dividing, and all of these divisions here are mitotic. That is to say, we're not shuffling genes at this point. We are replicating the DNA and creating genetic copies. And so we have germ cells that we set aside, and they are relatively quiescent until roughly around sexual maturity. Ovaries and testes will begin producing haploid cells that contain only 23 chromosomes. We call these haploid cells gametes. They are produced by this special form of cell division called meiosis. Meiosis is very, very similar to mitosis, but it has two splitting phases, and we're going to break out the mechanisms. We have our interphase, where we'll, G0 or G0, G1, S, G2, and then we'll have this first meiotic division. We call this Meiosis I. Then we're going to have a second meiotic division called Meiosis II. Now, the second meiosis is actually quite similar to mitosis, but we have some very, very special differences. You can see how the chromosomes have special patterning. We're going to go into all of those mechanisms. But the result of meiosis from a precursor cell is actually 4 cells rather than 2. Mitosis, the cell division product, is 2 cells that are genetically identical to the parent. In the case of meiosis, we have 4 cells that are not genetically identical to the parent and have only half the number of chromosomes, are haploid. Then those meiosis progeny, these gametes, can then potentially fuse with another gamete from the opposite sex to create a new diploid cell. So if we were to sort of break out the human life cycle, so here's fertilization here, and then we live roughly somewhere between 70 and 100 years or so on average. As diploid organisms now, once you reach sexual maturity, you will begin creating haploid cells, and so part of the human life cycle is as 2 independent cells. In testes, the sperm live roughly 3 to 7 days, something along those lines. Roughly about 3 days, 7 days at the most. So very, very short. With eggs, it's a little bit longer. Eggs are created, and then there are precursors of eggs that mature basically 1 or so at a time over the sexual lifespan of an organism. But even then, upon the forming of the final egg cell, that lifespan is relatively limited, especially when you compare the rest of the human life cycle. Well, we have fertilization, 9 months, and then the rest of that time. So it's a relatively simple life cycle. And our life cycle is almost identical to other organisms. It's just a question of how long the entire span is. For instance, an animal like a mouse, this whole span spans roughly between 1 and 2 years, whereas in a human being it could be anywhere from 70 to 100 years. In the case of plants and some algae, you actually have this sort of tripartite cycle. So we have fertilization here. We'll always start at fertilization. We create a zygote, and that creates a mature, what we call a sporophyte. It's a sexually mature diploid organism that has meiosis and will create spores. Those spores will live as haploids, and those spores can actually grow and develop by mitosis and create what we call a gametophyte. Okay, a gametophyte is a haploid multicellular structure that then will release cells that will undergo fertilization to create this sporophyte. So you have a sporophyte, you have a gametophyte, and then they fertilize to create another sporophyte. So you have different forms, whereas in mammals and in humans, you have a haploid stage where you have gametes, and then you are mostly diploid. In this case, you can have a period of time, a significant period of time in the cycle of this organism, where they are multicellular and haploid. Only unicellular and haploid in animals. Again, this is plants and some algae. In the case of things like protists, you can have a similar kind of a thing as what we see in the algae. Again, we'll undergo meiosis and create this haploid organism. It can be unicellular or multicellular, and then those can then fuse and create a fertilization that then creates a zygote in a diploid organism. So it's more complicated. We see this in fungi, okay? For instance, we can see this in mushrooms. So plants and algae have what we call an alternation of generations. That is to say, they are multicellular, but they are multicellular diploid, a sporophyte, and a multicellular haploid, a gametophyte. So plants and algae have a sporophyte and a gametophyte life cycle. The sporophyte, they are diploid. They create haploid spores. That's where sporophyte comes from. Haploid spores, those spores can grow and develop into haploid multicellular gametophytes that produce special reproductive cells that will cause fertilization and create a new sporophyte. So a diploid and haploid multicellular part of their life cycle. Diploid is the sporophyte, makes spores. Those spores can make gametes that then fuse to create a new diploid organism. So when the spore grows, it's growing by mitosis. It's called a gametophyte. It will make special reproductive gametes. The spores here are not sexual cells. They are not involved in sharing of genetic material. They grow into haploid multicellular organisms. Those haploid multicellular organisms then create gametes that are, shall we say, sexually viable for fertilization. Sporophyte, gametophyte. Sporophyte, gametophyte. Fertilization. Sporophyte, meiosis, gametophyte, making gametes, and so forth. In the case of protists, it's similar to the plants and algae, only the zygote, that diploid cell, doesn't ever go to a multicellular stage. They are only multicellular, potentially in the haploid form. So that zygote, really all its job is to create haploid cells by meiosis. So when we have that haploid cell, that meiosis created, you might grow into a multicellular haploid organism, or in some cases they will stay unicellular. Just depends on the species. That new meiotically created cell can then create gametes for fertilization. And again, you can get these sort of complex figure-8 type situations going on. I don't want to get too crazy about this. The main thing is that you have a diploid and a haploid phase in the life cycle. Look no further than mushrooms, like this very, very delicious morel mushroom. What this is a sporophyte. Okay, this is a sporophyte. It creates spores that then fly out and then create haploid gametophytes. Again, we have that zygote. It's only one cell when we talk about fungi and protists. Only one diploid cell. It does meiosis, creates those haploid cells, those gametophytes, that can create gametes that can then fertilize. And again, it can be haploid, multi or unicellular in the haploid stage, but it can only be unicellular in the diploid stage. So no matter how complex the life cycle, there are a couple of simple rules to remember. Mitosis is the only way that haploid cells can divide. So when you have a gametophyte making gametes, it's actually a special kind of mitosis. Diploid cells, we talked about somatic cells, divide only by mitosis. But only diploid cells, regardless of the life cycle that you have, can undergo meiosis. You cannot have haploid meiosis. And the point of meiosis and the point of sexual reproduction, as I mentioned before, is the shuffling, the random assortment of parental and maternal genes on the new chromosomes. You're going to have an equal number of genes that you had in the original cell. It's just that there's been a redistribution of maternal and paternal gene sets. And that's the point. Genetic variation. Genetic variation, the random assortment of traits designed to increase genetic diversity in the population. So let's talk about meiosis now. Things that they have in common, they replicate their chromosomes prior to the onset of the divisional process. In the case of meiosis, we actually have 2 cell divisions, and that's why we end up with 4 daughter cells. We have a Meiosis I and a Meiosis II. So we're creating 4 daughter cells rather than 2, and they're going to not be genetically identical. They're going to be genetically asymmetric. They have half as many chromosomes as the parent cell, and the sorting of the genes on those chromosomes is somewhat random. So when we look at this picture again, what we have prior to meiosis I is a gene exchange event within homologous chromosomes. In meiosis I, we call that reductional division. Okay, we're actually going to create haploid cells after meiosis I. So after meiosis, that's why we call it reductional, is because after meiosis I, there are only half the number of chromosomes in those 2 daughter cells. Now, in meiosis II, you're going to just split the chromatids. You're not splitting chromatids in meiosis I. So in meiosis II, we call that equational division, because what you're doing is you're doing just like mitosis, you're splitting the chromatids into separate daughter cells. Here, what we're doing is we're separating homologous chromosomes. That's why it's reductional. We're going from an N of 23 to a half of that, 12 and a half, shall we say. So we're going to have 4 daughter cells. The chromosomes are going to be asymmetric in terms of the genetic distribution. The interphase of a germ cell is very similar to the interphase of a somatic cell. What we have going on, we have a G1, S, G2 phase. G1, we are stimulated to grow and prepare for DNA copying. In the S phase, we copy the DNA, so now we have 2 sister chromatids. Here we have independent chromosomes, but we have duplicated the DNA in both of those chromosomes, and now they have 2 chromatids instead of just 1. That occurs in that S phase, just like mitosis. In meiosis I, we have that reductional division, so we're going to split the pair of homologous chromosomes. Let's say this is chromosome 21 in humans. One chromosome 21 is going to end up in one daughter cell, and the other chromosome 21 is going to end up in the other. The only problem with this picture is that these guys should not be all blue and all red here. There's another event that happens just prior to meiosis I that is critical for establishing genetic variation. But that's the reductional division. And then we have meiosis II, which we call equational division. This is almost identical to mitosis. You have the 2 sister chromatids, so you have the splitting of the chromatids, and then you have cytokinesis to create those daughter cells in both of the original daughter cells. We end up with 4 haploid cells. These cells are haploid here. These cells are diploid. So in meiosis I, we have the G1, S, G2 phase. Again, G1 is the stimulation and preparing for DNA replication. S is the synthesis part of DNA synthesis. We're duplicating the chromosomes. And then G2, we're preparing for meiosis. Now, when we start meiosis, the sister chromatids begin as genetically identical, but by the time that we get to the metaphase, those chromosomes will be genetically non-identical. So before we get to prophase 1, the chromatids are identical, but by the time we get to prophase 1, the prophase of meiosis I, they are genetically non-identical. You have that centrosome, okay? We're going to duplicate it, form two, separate them, just like mitosis, but there are some key differences. So in prophase 1, we're going to establish non-identical sister chromatids. So they start out as identical and then they become non-identical because what we do is we have this crossing over event. Well, the homologous chromosomes cross over and form what we call a chiasma. Chiasmata means plural, is a plural form of a chiasma. And we have a recombination event. It's called recombination, where we take DNA pieces and we exchange them for equal pieces, or they should be equal, on other chromosomes. So it's a recombination event, and now we have non-identical sister chromatids. Those non-identical homologous chromosomes are then going to sort along this metaphase plate, but instead of being single file like we have here in metaphase 2, they are in homologous pairs. They form 2 by 2s, and one chromosome move to this pole, the other chromosome will move to the other. So the duplicated chromosomes, the whole chromosomes move, not just the sister chromatids. And so we now have 2 daughter cells after cytokinesis that are haploid and genetically asymmetric. Those daughter cells will then undergo a second divisional step. This is the equational division. That's basically identical to mitosis, but again, the sister chromatids are asymmetric genetically. So even though they are splitting right down the middle of these chromosomes and the chromatids are moving, you can see that those chromatids are asymmetric in terms of their genetic material. Some of the material is from the father, some of the material is from the mother, and it's different ratios. So prophase 1, metaphase 1, anaphase 1, and telophase 1. Just like mitosis, the biggest difference is that we have this crossing over, okay? The formation of chiasmata, the formation of a tetrad, which is just 2 double chromosomes, okay? You get that crossing over event, the recombination of DNA, the shuffling of genes. Then they line up 2 by 2 rather than 1 by 1 on that metaphase plate. And that whole doublet chromosome is moving one chromosome to one side and the other homologous chromosome to the other. But that right there, that is one of the big reasons we have genetic variability. And this is true in any of the meiosis that you see in the entire biological world as we know it. This is the major mechanism of shuffling of genetic material. It's not just the mother and father creating a fusion. It's an asymmetric. So, for instance, you may have more genes from one grandparent than another, depending on the shuffling of genes. Prophase 1, metaphase 1. Again, what makes this different from metaphase 2 is that they line up 2 by 2 rather than in a single file. So we have prophase 1, very, very long time. That's because we're doing that recombination event. We're condensing the chromosomes, like packing our luggage. We're going to line up together. They call that synapsis. When you line up, again, homologous chromosomes line up, and then they form these crossing over complexes. You can actually see these things under electron microscopes. They cross over and begin swapping pieces of material. Very, very interesting molecular process here that's studied very, very closely. Why? Because we do see genetic anomalies. For instance, Down syndrome. We call some of these things non-disjunction. That is to say, they formed a crossover and then they couldn't separate properly. So we form this chiasma and then we recombine the DNA into asymmetric chromosomes. So the non-sister chromosomes exchange segments of DNA. When they are crossed over like this, you have 4 sister chromatids. That's where the word tetra, 4, a tetrad. So when you have these X regions, that's a chiasma, and then you can have multiple crossing overs or chiasmata. So let's look at this just in an animated form. So we have two parental chromosomes, okay? Your autosome from your mother and your autosome from your father. In prophase 1, they line up, and this is called synapsis. Then they will form these crossover things, and that's called a chiasma, or in plural, chiasmata. They will recombine their DNA, that is to say, exchange material, and the tetrad will then separate. So we now have recombinant DNA, and during metaphase 1, those tetrads will now separate to different poles, being asymmetric. So the tetrads line up on the metaphase plate, and each chromosome moves from one side to another. And again, it's not going to be all mother on one side and all father. They will form up, and maybe some of the that are predominantly maternal will come over here, along with some that are predominantly paternal. But the microtubules, the kinetochore, is an identical mechanism, only that you're moving a whole chromosome versus a chromatid. In anaphase 1, we are moving the whole chromosomes, one homologous chromosome and another homologous chromosome, instead of chromatids. That's the only real difference between that and mitosis. Telophase 1 and cytokinesis, there's no real major difference mechanistically, only that the daughter cells are now haploid. They have doublet chromosomes, but they are haploid. And just like in mitosis animal cells, you form a cleavage furrow using actin microfilaments. In plant cells, you have a cell plate forming of a cell wall. Between meiosis I and meiosis II, you're not going to have any replication. Another major difference. Unlike in mitosis, you should have replication before you divide again. Meiosis II is virtually identical to mitosis. Prophase, just like mitosis, only you're not condensing the chromosomes this time. They're already condensed, okay? So you don't have another replication of DNA, and you don't have another condensation event. What we are going to do is we're going to organize those chromosomes, single file, on the metaphase plate, separate the chromatids, and undergo cytokinesis. So very similar to mitosis. And here are the steps you can see from your book. They line up like this, and note that the chromatids are asymmetric. Some have some different ratios of parental DNA in them, and when they split, they are going to be non-identical daughter chromatids. So I'm going to stop right there for now, and I will pick up the rest of chapter 13 in part 2. Thanks for your time, and as always, if you have any questions, please just let me know, and I will talk to you again soon.